This conference will now be recorded.
Today we are honored that DNA Day is global and international, and pleased to partner with H3Africa to bring you researchers at the forefront of genomics. I have a few housekeeping items for participating in this webinar. As you just heard, the webinar is being recorded for future sharing purposes. Please mute your microphones so as not to speak over or interrupt the speakers. All questions and answers will be done at the end of the second presentation. If you do have a question, please type the name of the speaker it is for and then your question in the chat box. And finally, it is my honor to introduce the National Human Genome Research Institute's Dr. Carla Easter.
Good morning, everyone, and thank you so much for being part of this historic program. This is the first time that we have actually worked with H3Africa for DNA Day, and we're incredibly excited. It's my honor and pleasure to introduce our first speaker for our exchange, Dr. Ayo Doumatey. Dr. Doumatey received her bachelor's and master's degrees in biochemistry with minors in microbiology and cellular and molecular endocrinology from the University of Claude Bernard in Lyon, France, and earned her doctoral degree in microbiology and immunogenetics from Howard University in Washington, D.C. Dr. Doumatey is currently a staff scientist at the Center for Research on Genomics and Global Health at the National Human Genome Research Institute, where she has spent the last decade conducting research on the genetic epidemiology of metabolic disorders, especially obesity and obesity-related traits, including type 2 diabetes in populations of African descent. In addition, Dr. Doumatey has been a wonderful colleague and friend to our outreach program, and it's my pleasure to introduce her to provide our first presentation.
Thank you, Dr. Easter, for the nice presentation. And without further ado, I will start my talk. The title for my talk is Understanding the Molecular Profile of Metabolically Healthy Obesity in African American from Single Molecule Approach to Omics. So this is the outline of my talk. I will first go through some housekeeping information on obesity, and then really focus on a subtype of obesity known as metabolically healthy obese individual. And the talk is kind of split in two. I will start with a hypothesis-driven method to look into how this subgroup has defined our thinking about obesity and obesity research. And then I will go into a global approach where we don't have any hypothesis, but we just screen a bunch of protein DNA to see how that is related to the phenotype. So as we all know, obesity has been an epidemic, and it has not just been affecting developing countries, but also low and moderate income country. So I really like this cartoon because it shows us how in the 90s, as we were skiing, moving toward the year 2000, we start gaining weight. The TV starts getting smaller, but we as humans start getting bigger. And that has a lot of consequences metabolically, and this is basically due to accumulation of fat that is actually a consequence of positive energy. So basically, a lot of people have been eating more than they exercise. So with time, the excess nutrient accumulate into the fat cells. So obesity is actually a really complex trait, which is characterized by excess adipocytes, and also the consequence is that you develop a lot of comorbidity such as type 2 diabetes, hypertension, and cardiovascular diseases. Those diseases actually cost a lot to insurances, and in the United States, for instance, is in the billion of dollars. And as you can imagine, in US, for instance, obesity is not actually spread through the population equally. African Americans tend to have a higher prevalence of obesity, and 2/3 of African Americans are either overweight or obese. In epidemiology study, the easiest way to measure obesity is through BMI, which is easy to obtain when you're dealing with a high number of people you're screening, and it's basically using weight and height. There are different types of obesity. Not all obese are equal. So depending on where your fat is accumulated, you will have different outcome. So it has been shown that people that accumulate their fat under the skin, tend to be healthier. But on the other side, people that accumulate fat below the waist tend to have a more complicated outcome. Most of them will go on and develop type 2 diabetes or other complications. So those two obesity are known as apple shape or pear shape. The pear shape obesity tends to be more frequent in women where they accumulate their fat really around the waist instead of in the abdomen. So when we look at the different type of distribution, here the scan is showing the subcutaneous, which is less pathogenic, and then the visceral. The visceral basically, once the fat cells themselves are full of triglyceride, they have nowhere else to go. So they start accumulating around key vital organ, especially the liver. So obesity not only is complex, it affects almost every single organ of the body, as you can see on this slide. So from the brain all the way to the bone system. So it's very important that we understand the molecular basis of obesity so that we can address this epidemic. So not only you see the obesity from the outside phenotype, as I just showed in the other slide, but there are also a lot of things happening cellularly. So if you take an adipocyte, an adipose tissue, when somebody is lean, you have very few immune cells. As the person starts gaining weight, you have this storm of cytokine that has started being released by the adipose tissue. A lot of them are actually pro-inflammatory cytokine, as you see here, TNF-alpha, IL-1 beta, and IL-6. And one that is really key here is adiponectin. It's one of the few adipokine that is inversely related to BMI or fat. What does that mean? Basically, every other molecule that is released from adipose tissue increase as one gain weight, but adiponectin actually is decreased as you gain weight, and the reason is because adiponectin is an anti-inflammatory molecule, and I will spend some time talking about that. So in the normal course of somebody that is obese, the progression of the disease is you gain weight, and most people will move to a metabolic syndrome and then eventually develop type 2 diabetes. And along with that, they may develop cardiovascular disease or fatty liver. Now, we have a small subset of obese individuals that will not, it will take them some time to get to develop type 2 diabetes. And there are some that have been reported to be overweight or even obese all their life to their late 90s without even developing any of these conditions. So those are the individuals that I have focused my research on, because looking at those individuals in the time frame where they do not develop any complication, it will give us a better understanding of the pathophysiology of obesity and maybe even target a drug to those things that we can find. So how do we define metabolic healthy obese? It's basically people that have a BMI greater than 30, they have a fasting glucose less than 126, they have a healthy blood pressure, which is 130/85, and they have a really good lipid profile, as you can see here. So these two pictures are showing the prevalence of the two phenotypes. On the left side, the unhealthy obese represent about 70% of the obese population, and they tend to have a bad outcome. Whereas in the metabolic healthy obese, 30% of them, of the obese population, tend to stay healthy for a longer period of time. It may be a transient a healthy state, but again, some people stay healthy throughout their obesity time. So basically, the bottom side is just showing that these individuals usually will not move toward type 2 diabetes. So what is the problem? There is no consensus method of identifying MHO. The definition that we've been using is basically using variant of metabolic syndrome definition. And as many of you will know, those definitions vary. And it may not be a good criteria to identify these individuals. 2, 3, they do not respond favorably to lifestyle change. So there was a big study conducted in Canada where they put a group of obese on lifestyle change, including diet, and a lot of the individuals that were defined as MHO, actually, even though they lose weight, their outcome did not change. So basically, if you put somebody that is defined, some of them that are MHO on diet, they actually don't really, you don't really see the benefit of those life change. So that tells us that you cannot apply a diet regimen equally across obesity because you have different subtypes. So the molecular basis of MHO, of course, remain elusive, and that's why we are conducting this study to better understand what's going on. So I showed this slide earlier, but what I want to point out here is that we know that high adiponectin is actually protective, and you usually only see in lean state. But the question now is to see if an obese person, for instance, if they're healthy, is it that the adiponectin remain like a lean individual? So that's the question we pose in one of our studies. So what we did is to look at about 800 individuals. They're all unrelated and they're African American. We measure total adiponectin using ELISA, and we just look at association between the MHO phenotype and adiponectin. So this is just to show you basically the characteristic of this individual. You can see that the main difference between the gender is that women tend to be more obese compared to men, but they actually tend to be healthier, as you can see in the number of the adiponectin. This can be explained by two things. Women tend to have more fat, therefore they tend to produce more adiponectin, but also the fat is accumulated around the waist, not in the abdomen. So that tends to be protective in women. So in this study, the prevalence of obesity was 42%. And women, as I said earlier, have a healthier profile compared to men. And one of the reasons is because of the distribution of the adipose tissue, and therefore they produce more adiponectin. So here we look at, among the obese, we look at the prevalence of how many people have higher adiponectin. And then it comes out to be 28% of individuals actually have higher adiponectin despite the high weight. So in that case, that's unexpected. So therefore, we call it paradoxical hyperadiponectemia, because you're not supposed to see that much high of adiponectin in somebody with the kind of weight that we see in here. And how did we define that paradoxical hyperadiponectemia? We just use the cutoff in the lean in this population. So for instance, in men, for somebody to be to have a higher adiponectin, you have to have a adiponectin level greater than 8,000, and in women is greater than 11,000. And doing that, what we saw is that if you do a regression, a logistic regression, individuals that actually have higher adiponectin are 2 to 3 times more likely to be metabolically healthy. So again, the prevalence, when you apply those definitions, the prevalence of hyperadiponectemia among those who are healthy is 19%, and 42% of the people that have high adiponectin are also metabolically healthy. So now, instead of just looking at one molecule, we know that in obesity, there are many molecules that are actually released. So what we did is to use a very strict definition. We pick all the different definitions that have been used in literature. And before you can be in this set of study, you have to be defined as metabolic healthy by all 3 definitions. So when we do that, we end up having to include in this study 20 individuals, 10 healthy and 10 unhealthy. And because the prevalence is higher in women, it ends up being all women in this particular study. So again, these are the definitions. So the definition as shown earlier is the basic definition, which include hypertension, diabetic, and lipid profile. And the definition 2, which is a variation of 1, is just that you're changing the cutoff to be more stringent, where you actually taking away anything that can even potentially be pre-diabetic or even pre-hypertension. And then the third definition is just adding the inflammatory component. So you have to actually be classified as metabolic healthy by all 3 definitions to be in this particular study. And this is the chart of how we get to it. We take the intersection of those 3 and we end up with these numbers. So these are the characteristics, and as you can see, these 2 groups are not really different except on, again, waist hip circumference, as I was telling you guys earlier. So the MHO tends to have a lower waist circumference, and then the adiponectin is higher in those individuals. So here is the volcano plot. So we screen about 11,000 protein in the serum of this individual, and only focus the analysis on 7,000, because those 7,000 are the ones that were detected in both groups. And when we do that, we actually found 56 proteins that are differentially expressed between the 2 groups. And after correction, 20 of them remain significantly differentially expressed. So I have a table here that shows those molecules, and it's quick glance at these molecules show that a lot of them actually fall in the inflammatory pathway. So most of the protein that are underexpressed are actually pro-inflammatory protein. So pro-inflammatory protein are underexpressed in healthy individuals compared to unhealthy individuals. And then lipids classes are also overrepresented in MHO individual. So here we can see, moving from one molecule where we say adiponectin, an anti-inflammatory molecule, is overexpressed. Here is confirming that inflammatory is playing a key role. And in addition to that, this study is showing us also that lipid may be an important part of this process. So I already talked about all of this, so I can move on to the next slide. Just having a list of protein really does not tell you which pathway these molecules are actually operating. So doing a IPA analysis, we can see, as I said earlier, that lipids and inflammatory pathway are the one actually driving clearly this phenotype. And here you just put all of these molecules in each of the pathway, and again, inflammation is downregulated, and lipids tends to be optimally efficient in this individual. So the next question is, can we use those 20 differentially expressed protein to actually classify this individual? And when we do that, applying to a larger cohort, we can see a clear separation in the two groups. So this is actually a set that can eventually be validated to actually detect this individual. And again, this has been done in African Americans, so it have to be confirmed in other populations. So the summary basically is anti-inflammatory are overexpressed, lipids are efficient in this individual, and the mechanism by which that happens is because this individual tend to actually maintain the fat inside the adipocyte. They're not really releasing it into the circulation so that it can accumulate around organs that are effectively involved in insulin metabolism. That's the take-home message from this study. So secondly, we know that protein are actually regulated by other mechanisms. So what we wanted to do in this study is to see if microRNA, for instance, which is really known to regulate protein level, are playing any role in what we found in our proteomic study. So basically, it's a follow-up study. We use exactly the 20 people screened for 179 microRNA, and this is just to understand the mechanism involved in the regulation. Why microRNA are alluded to that? They're small non-coding RNA, 22 nucleotide, and they tend to bind to the 3' untranslated region of the messenger RNA, encoding for a particular protein, either by degrading them or by repressing their expression. So when we screen those 179 microRNA, this has been done by RT-PCR. I will not go into the technical detail due to time constraint, but what I want you guys to know is that doing that, we find a lot of microRNA that were either overexpressed or underexpressed, but after multiple correction, we actually positively identify 8 differentially expressed microRNA in this set. And the next step is to validate these 8 in a larger cohort of 100 African American. And again, what we see here is that one party of the 8, only one remains significantly associated with all the phenotype that we look at, and that's the microRNA 374 5P. And here, I'm showing one example of the outcome that is driving the change in this microRNA level is TG over HDL, which again are lipids and also known to be associated with insulin resistance. So it seems like metabolically healthy individuals actually have low TG over HDL, while they have an overexpression of microRNA 374. So the next thing is to see the target of this microRNA. So this can be done in IPA ingenuity. So what we found is that this particular microRNA is able to target the regulation of 37 messenger RNA. But because we know that the microRNA that are released in obese are actually released from adipose tissue, so we restrain the analysis to just adipose tissue. And when we do that, we go for 37 microRNA to just 3 microRNA. Now, what do we do with that? What we did is to take those 3 messenger RNA that are targeted by this microRNA and to validate it in completely independent cohort of African American, and we the EN1 didn't pass quality control, so we end up only analyzing CCL2, which again is a pro-inflammatory molecule in step 4. And here, CCL2 is the only one that remains highly significant in this population we use for validation. So the trend in the mechanism that we're finding is still continuing, is still inflammation, is still lipid. And you can see that just using 3 different methods, we are picking up the same signal. So that tells us that inflammation and lipids are main key mechanism protecting this individual from developing comorbidity. So this are the conclusion which I just said earlier. And basically, inflammation is a big part of protecting this individual from developing metabolic disorder, and also lipid is playing a great role. So that's basically the take-home message. So one can think of maybe if we target those 2 main metabolic pathway, we can actually help other obese that move on to develop type 2 diabetes. So for those who are interested in continuing reading about this, we also done a lot of transcriptome analysis in this individual, and the conclusion is basically the same. We pick up other pathway, stress in ER, which are basically also again inflammation. So with that, this slide concludes my talk, and I want to acknowledge all the people in my lab, Dr. Easter and Dr. Hall, for inviting me to give this talk, and for Ms. Wise for organizing this whole symposium. Thank you.
Okay, thank you so much, Dr. Doumatey, for that interesting presentation. We'll only do questions at the end of the webinar. For now, I'm kindly going to ask Professor Nikki Mulder to introduce our next speaker for the day, and then we can obviously proceed with the rest of the webinar. Thank you so much, Nikki. Go ahead.
Thanks, Pablo. And I also want to express my welcome on behalf of H3A Bionet to this symposium, this mini symposium DNA Day. We're very pleased to be partnering with NHGRI and this at NIH to have the fellows together. So I will proceed then to introduce the next speaker. So from the H3 side, we have Jorge da Rocha. He's a PhD student at the Sydney Brenner Institute for Molecular Biosciences at the University of the Witwatersrand in South Africa. His work is focused mostly on pharmacogene variation analysis, particularly in African populations. So he's built up a lot of expertise in variant annotation and functional assessment of variants using various different tools, including bioinformatics. So he is he's the Young Researchers Representative for the South African Society for Human Genetics. And he has a passion for working with young people, and I think he's really interested in having further interactions with this fellows group. So he's going to speak to us on an ADME project that he's been doing in African populations, entitled The Extent and Impact of Variation in ADME Genes in Sub-Saharan African Populations. So thank you, George.
Hi, everyone. Just mic check. Can you hear me?
Yes, we hear you.
Oh, wonderful. Right. So thank you very much for inviting me here today. It's great to participate in DNA Day. I'm going to be presenting this work on behalf of a really big collaboration. It's quite a wide one. We work quite in depth with H3AFRICA fellows, and we're also supported by GlaxoSmithKline. And we're quite broad in that we have universities all across Africa helping us. We've got WITS, we've got UCT, we've got University of Khartoum, and we've even got colleagues from the University of British Columbia up in Canada. Great. So I'm going to be telling you about the work we've done to try and characterize ADME variation in African population. These are genes that may influence drug response from their pharmacokinetic sides. And to do this, we started off by deciding, okay, what do we want to do? We want to get a broad assessment of these genes in our data sets that we have available. And we needed a set to select. So we used a list defined by Farm ADME, 294 ADME genes that we could use to mine. 32 of these genes are core genes, and 272 are extended genes. And these are based on how many known biomarkers there are for pharmacogenomic effects. Core genes are those with the greatest number of biomarkers and significant evidence that they're important in pharmacogenomic roles. How we did this analysis is we tried to use as many diverse techniques as possible. We don't have any phenotypic information for these populations because we put them together from our collaborators' data sets. So we're trying to use more data analysis techniques based on the data alone. We started off by just characterizing overall variation, looking at copy number variants, really focusing in on coding variation within these genes, seeing how many of these variants are novel, slight but on regulatory variance analysis, and then we did this all by comparing African cluster populations together. So just a summary of which data we used. We have a large collection of high coverage whole genome sequencing data. This was provided by H3A collaborators. In total, 458 samples were brought together. 412 of these were novel. They have not been studied in this way before, and 46 were provided from previous work. We have really good numbers down south in Southern Africa, and a nice cluster in Western Africa, with a few sparse numbers from the other data sets which we could bring in. To start characterizing this, we ran a principal component analysis on these, including some data we had from the H3AFRICA genotyping array. And we decided to try and visualize these on a PCA, and we see that we have overall broad representations of Southern Africa, South Central Africa, Western Africa, and Far West Africa. Now these circles that I've drawn here are just diagrammatic. We clustered the samples with k-means-based clustering using our mass. Our first point that we'd like to investigate is high impact variance in terms of coding variation. Which of these, based on their genomic characteristics, are likely to be damaging to their gene function? So we use the model that's predictive for ADME variance. It incorporates 5 genomic functional prediction tools. We can also look at loss of function variation using these. And in total, we identified 930 high impact variants. Most of these are quite rare, below a minor allele frequency of 1%, and 80 of them are novel. So looking at how these are distributed across the African clusters, I've got Southern Africans here in blue, South Central in red, Far West in green, and West African in gray. We see that if you put all of these together and see how many overlap between more than one cluster, only 104 of the 930 are common to all 4 African clusters, whereas either similar or greater numbers of variants are specific to each cluster alone. And these tend to be underneath that minor allele threshold of 1% indicated in this red line over here on chart B. Looking at how the distribution of those unique, well, let's say not unique, discrete variants to each population are distributed. Southern Africans have the highest frequencies of these overall, with all the other clusters generally having only rare variants discrete to them alone. Visualizing the most common high impact variants on a heat map chart, what I'd like to show you here is that if you compare the frequencies of these within their relative clusters, you don't have an overall even spread of frequency. So for example, in Southern Africans, you've got a variant over here that is roughly 20% minor allele frequencies that's completely absent in West Africa. And as the frequency tends to be more common, you never see a straight color across the board, meaning that even at the most common variants, not all populations will have the same frequency for a given variant. We then decided to do some intercluster comparisons with FST to measure, are these significant differences between the populations? And what we've done here in chart A is taken our high impact variants and we've extracted them out from each group and calculated FST scores for these. What I've shown on the chart here, for example, West Africa, there's a central node represented by the geographic midpoints of where all the samples in this cluster came from. Overall, we see the strongest difference for high impact variants between far West Africa and the Southern African cluster, whereas very small differences for West Africans and South Central Africans, which we found quite interesting. On chart B over here, we haven't shown the map because this is the representation of overall ADME variation, not just high impact. And though we do see that there are, of course, the pattern reflected larger differences between Far West and Southern Africa, these differences are not significantly different from non-ADME genetic variation at all. So for ADME higher impact coding variants, some of these differences are significant, but for ADME overall, the variation there seems to be based on genomic variation overall. So looking at transferability of ADME variants that we have strong known clinical effects for, we've plotted some of the key variants over here for known pharmacogenes on the x-axis. And what's resounded with us is that a lot of these variants, although being so well characterized, are quite rare, not very common at all in African populations. For others, we see that the groups don't tend to differ too much, the four clusters together, although you can see differences of about 10% in minor allele frequency between them. Looking at how many individuals in each cluster carry at least one of these clinically usable variants, we see that on average between 5 and 6 variants are carried by each individual in each cluster. So we decided to look at novel variation too. We have these data sets, so we wanted to know how many of these variants have not been even reported before. And what we've got here are these circus diagrams which try and show which ones of novel variants are connected between more than one group. So I'm going to focus on this West African group over here on figure C. We've arranged the variants in order of most common minor allele frequencies to most rare, according to this index for all charts. And what we see is that even at common frequency levels, you don't get complete overlaps between all four clusters for these variants. You tend to get sparse, spotty overlaps. And for C is the high impact variants only. And for total ADME variation, you see it a more complete pattern that at more common levels, the variants tend to be shared. But once again, there is a vast amount of novel variation that is only found within a single population cluster. Having a look at some of the structural impacts that can happen from these novel ADME variants. So we see that most novel ADME variants tend to occur in the cytochrome P450 family. And on visualizing some of these variants, we see that they can affect really important groups within their protein structures. And the same goes for the variants within the CFTR, ABCB1, an important drug transporter, and NAT1. And so we think that some of these novel variants may have significant structural consequences and thus significant impact on drug response should these proteins be critical for what that drug needs to do or move to or be metabolized by in the body. Just overall, some conclusions. What we've understood is that the ADME landscape in African populations is significantly diverse. and that rare variation, which is often population specific, could play a large role in ADME-related effects. African clusters are distinct from each other, and geography isn't an ideal proxy to be used for how vast genetic variation can be. And we encompass this problem we term the genetic diversity bottleneck, which is when you want to put into place pharmacogenomic interventions or strategies, you need to consider how genetically diverse Africa is. And the more populations you study, you will see that complexity going up. There's definitely going to be strong limitations for using technologies such as genotyping arrays. Arrays can only contain known variants, and we see with our whole genome sequencing data that there's a lot of novel variation that could have a strong effect. Obviously, whole genome sequencing and whole exome sequencing would be better, but once again, you need to consider the strategies you employ for common variants and rare variants. Both of them will matter for your approach. And that based on for variants, we have a lot of evidence for. Current databases tend to have an evidence bias towards Europeans for pharmacogenomic effects. What we need there is more pharmacokinetic and clinical trial work in African populations to understand which African variants may have the strongest clinical effects. And overall, definitely, we need more sequences and pharmacogenomic studies before we can implement pharmacogenomics across the African continent. We finished our first paper. It's currently with our collaborators, and we're doing a lot of review. We're hoping to get this submitted soon, and we're really excited about it because it will be the largest in-depth study of ADME variation in Africa so far. So hopefully all goes well there. And I'd just like to acknowledge the great team. This was not done in isolation. This was a really nice collaboration between African researchers, but are mainly based on the African continent. And we have some fellows overseas, like our colleagues in Canada, who provide us really good advice and help us unpack the meaning of our work. So thank you to everyone. Thank you to H3A Bionet, and thank you to you for listening today. I know it's not an easy time during the time of COVID, but it's great that we could all join together and share this work. So thank you very much.
Okay. Thank you so much, George, for another interesting presentation for this joint webinar. As the Ed said, we're supposed to have 3 presenters, and I want to check to see if the 3rd presenter has managed to join us. Her name is Busi Mlotra from the University of Botswana, who's going to speak about TB and HIV effects in African populations. Busi, are you in the audience by any chance? And are you able to speak? Busi? I'm gonna give it like a few seconds just to see if anyone replies, because I see there's a lot of calls with anonymous names. Busi might be one of them, so I'm just gonna check. Okay, so I'll actually, in the interest of time, apologize on behalf of Busi. She's currently in an area where there's low or no connectivity as well, and we were hoping that she'd be able to make the joint webinar. However, as I've given it a few seconds, it seems as though she's unable to join. So apologies on that part. Technology and with the whole crisis that's going on globally, it was beyond our control. But I'm sure we'll get to see and hear Busi present in our future presentations. For now, I just want to say thank you so much to Dr. Doumatey and to George for your presentations, respectively on ADME genes and obesity in African American communities. I'm sure there's a lot of questions that we have from our audience. And as Roseanne said in the beginning, when welcoming us, if you do have a question, kindly type it in the chat box. Rolanda, which is one of our co-organizers, will kindly fill the question for us. So if you have a question, a burning question right now, kindly do raise your hand or do type it in the chat box, and Rolanda will go ahead and read it. We have about 15 minutes to go to either comment or ask questions to our speakers. So please go ahead and type the questions.
While we're waiting on some questions, I just wanted to encourage you. The reason that we set the this webinar up in the way that we did, where people entered their bios and information about them, was that we'd like to encourage the different fellows from Africa and the US to get together, have discussions, to match yourself up with somebody who's doing something related to you. So you're also welcome to give a shout out to somebody if you noticed their bio and you think that they've got some interesting research and you'd like to chat to them further. Feel free to shout that out now. And also to contact them afterwards. We have created a channel for you to have discussions. So the idea here is that this is an introductory forum where we hear some great presentations, but then we also get that you guys get chatting about your research afterwards. So we want to encourage that. And everyone's obviously being quite shy about asking questions.
Thanks, Nikki. So we do have one question from one of the speakers to the next speaker, in fact. Ayo wants to know to George, can the new H3A chip be used for pharmacogenetics?
Oh, this is a really good question. So in our brief look, we have seen that there's only about a 5% coverage of the chip of the variants that we've identified. So for some variants that are very strong and impactful, like for example on the G6PD gene, we have used the chip data to unpack some of its findings, and not in this work, but in work to come. But for overall pharmacogenomics, I think even that chip will be limited. So you'll be limited to common variants that have known effects. Of course, future chips or future assays might be better once we figure out the best way to design them. Thank you.
Sure. Thanks, George. Others, please. Do you have questions? More questions?
Hi, Roland. It's Michelle. I just wanted to ask George. I'm not sure if he spoke. I came in late. I'm not sure if he spoke again on the tool that they've developed to basically input their variants and combinations of variants. I just wondered, what's the future of that tool? Do they anticipate that it will be improved or you know, modified or anything like that in terms of the data that they've got and they anticipate getting in future, and how widespread do they think it will be used? Will it actually be clinically used, or yeah, maybe just comment on that. Thanks.
Sure, that's a great question. So what we've done is we've made a Nextflow workflow, which we've called Gminer. Our code is up on GitHub. It's private for now, but it will come out with the paper. What it does is it combines a few pre-existing structural bioinformatics tools that you can then input a variant call file. It does some minor QC for you, and then it runs functional prediction. So that's free to share with everyone because we've used tools that exist in the public, and you just need to configure the databases yourself. One of the problems with this is that it's got a large, it has a large database component to it. So we are looking into alternatives in which we could package into a Docker, and that could be much widely used because it's just a one download, easy to set up, and your package comes with all the data you need to run your analysis. On whether it could be clinically applied as current, I don't think so. What this tool does is it's good for highlighting genomic impacts, but ADME variation is a bit complex, so we would have to add a few steps there to try and get which is the most usable information based on clinical evidence before they could be clinically applied. But these are the baby steps to that regard. We'd love to see it get to a point where we could do that. Thanks for the question.
Thanks so much, George. So we have a few more questions from Issa. He says, thank you for the great presentations. He has two questions for George. On the intercluster comparison of variants, I realized the node sizes were a little different. Is that supposed to have some significance? That's question number one. Do you want to comment?
Sure, I'll answer that one quickly. Yeah, we've done our best to correct for population sizes where our clusters are different sizes, but we've aimed to keep them at similar numbers where we can. We've also supplemented some of our analyses with data from 1,000 Genomes Project, where we have fewer samples in regional clusters. So that's helped us to do that.
Thanks, George. And Esther's next question for you would be, would you expect to find a similar pattern or distribution of these ADME variants in African populations genome-wide?
Oh, yeah, absolutely. So we saw that when we did our FST. So we do think there's significant variation genomically, so genome-wide. ADME variants, ADME genes are defined not by their particular evolutionary families or origins. They're defined by, we have drugs that are known to interact with them, and that's how we pick them up. So, of course, the more we learn about the genome, about the features of such genes, I do expect there to be more variation. Yeah.
Thanks, George. And the next question is for Ayo Doumatey. How will your research impact how we think or rethink obesity, knowing that there are individuals who have protective variants?
Can you please repeat your question?
Sure, Aya. So it's from Carla. She's asking, how will your research impact how we think or rethink obesity, knowing that there are individuals who have protective variants?
So basically, those individuals are helping us actually understand the overall pathophysiology of obese as a whole. So knowing that, knowing those variation in the subset can actually help target intervention in the overall obesity. So basically, that's the end rule. I don't know if that's the question the person was asking.
All right, we'll move to the next question. It's also for you, Ayo. It's Belen Hill asking. There is an obesity epidemic in children, at least in the US. Do kids tend to accumulate fat in apple or pear fashion? Does it relate to gender?
So depending on ethnicity, so like if you look at Hispanic in US, they tend to have an apple-shaped obesity, which is the one that is most pathogenic. The same is true also in Native American. However, in African American, I would say about a good proportion of African American, especially African American women, tend to have a more subcutaneous adiposity, which is less pathogenic. So it really depends also on ethnicity and also on gender. Men, the prevalence of MHO is lower in men overall across ethnicity because they tend to accumulate fat in the stomach area.
Thanks, Ayo. And then there's another question for you from Merit. He's asking, is MHO hereditary?
So that's a really, really good question. We have tried to address that, but you really need a family structure to look if it's a trait that is goes through family. But unfortunately, we didn't have enough power to actually answer that. So hopefully, as we accumulate more data, it will be something that we probably will be able to answer. But as to now, we have not been fruitful on showing if it's is transmitted through family members. But it's really something that we have in mind to see.
All right, he's saying thank you for that. Yeah. Are there any other questions? Well, in that case, we have 5 more minutes to go. So if there's any burning issue, you're also welcome to unmute yourself and speak if you should want to do that. All right. If there's no more questions, yes. Somebody wanted to speak? Okay, so if there's no more questions, I just want to take the opportunity to thank everybody that has worked very hard on putting this mini symposium together today, especially to Roseanne, Carla, Belen, Babalu, Nikki. Let me just see, there might be another question. So somebody's quickly asking, Mariba, I'm not sure to who this is direct. Oh yes, it must be directed to Ayo. Is there a test available to diagnose adiponectin levels at the moment?
Okay, so you can measure total adiponectin using ELISA. There are commercially available kits for that from different vendors. So total adiponectin is the easier one to measure, but you can also measure the different subtype of adiponectin, and you also have a kit available for that. And the kit is about, I think, $500 to measure adiponectin level in about 96 individuals. If you use a multiplex approach, is a little bit cheaper, but of course you have to have also the equipment that you will use for that. But yeah.
All right. Thanks so much, Ayo.
No problem.
So then I think that is the last of the questions. I'll continue. I just want to thank again the organizers, which is Roseanne, Carla, Belen, Babalu, and Nikki, and then also to you, the speakers, even Lucy, who could not join, sadly couldn't join us today, but to George as well, who stepped up to also be a presenter today, and to you, Ayo, as well. Thank you very much. We are very grateful that you could take the time and the effort to put these presentations together. So
thank you.
I don't know if there's anything else from anybody else that'd like to say something.
This is Roseanne Wise, and I echo all of your thank yous, and thank you to you as well, Rolanda, for your help with all of this. And we look forward to future collaborations. Thank you.
Yes, you're welcome. Thanks everyone for joining. And then goodbye.
Goodbye.
Thank you. Thank you
Thank
so
you.
much.
Bye.
Thank you.
Thanks, everyone.
Bye.
Thank you.
Bye.